You know, cancer is one of the biggest, most complex puzzles in all of medicine. We tend to talk about it like it's a single thing, but the reality? It's way, way more complicated than that. But now, there's a groundbreaking new approach that's letting us read the hidden story inside every single cancer cell. And believe me, it's changing everything.
I mean, this is the question that keeps doctors and patients up at night, right? You see a patient respond to therapy, the tumor shrinks, things are looking up, and then, months or years later, it's back. And often it's more aggressive and resistant than it was before. To figure out why that happens, we've got to look deep inside the tumor itself.
And this really gets to the heart of the problem. We think of a tumor as this single, solid enemy, but it's not. It's more like a bustling, diverse ecosystem of different kinds of cancer cells. So think less single monster and more chaotic crowd, with all these different groups pushing and shoving, each with its own agenda. Now there's a scientific name for this, of course. It's called clonal heterogeneity. All that means is that inside one tumor, you have multiple distinct families of cells, or clones. Each of these clones has its own unique set of genetic mutations. And it's this very diversity that gives cancer its power to evolve, to progress, and to outsmart our very best treatments.
For years, we've known this was a problem, but our tools just couldn't keep up. Our old methods could only track one mutation at a time. That's like trying to understand that whole chaotic crowd by just listening to one person shouting. Plus, these tools needed fresh or frozen tissue, which meant this massive historical library of patient samples was basically locked away from us.
But that is all changing. And I mean really changing. There's a new technology called GoT-Multi, and it's giving us a revolutionary new lens to see inside these tumors with a clarity we've just never had before.
Okay, so here's the genius of GoT-Multi. For thousands of individual cells, it sees two layers of information at the exact same time. It reads the cell's permanent genetic blueprint, that's its genotype, and it reads its real-time activity log, its transcriptome. It's like the difference between just having a list of ingredients in a kitchen versus actually seeing what meal is being cooked right this second.
So how does this whole thing work? Well, you can break it down into three big steps. First, it uses these special probes and some pretty smart machine learning to figure out the unique genetic signature of every single cell, sorting them all into their proper families or clones. Next, it builds a family tree for all those clones so we can see how the cancer actually evolved. And finally, it reads that activity log for each clone to figure out what it's up to. And this process gives GoT-Multi three incredible superpowers. It can track dozens of mutations all at once, not just one. It uses AI to make sure those genetic readings are super accurate, and, this is crucial, it's compatible with preserved tissues. We're going to come back to that one because it is a huge deal.
Okay, but theory is one thing. Does it actually work in the real world? Well, researchers took GoT-Multi and threw it at one of the toughest challenges in cancer medicine, a deadly, therapy-resistant lymphoma. This particular cancer is called Richter transformation. It's a really aggressive lymphoma that actually evolves from a much slower-growing leukemia. It often shows up after therapy, and honestly, the prognosis is usually pretty poor. It's the perfect test case for a cancer that's a master of evolution and resistance.
And here's what they found. It's incredible. Using GoT-Multi, they could map out the tumor cells not just by their genes, but by what they were doing. They sorted the cells into these different neighborhoods based on their activity. You had some cells that were multiplying like crazy, others that were stirring up inflammation, and another group that was in a full-on stress response mode.
And this led to a discovery that kind of turns our old thinking on its head. This quote from the study says it all. Basically, they found that genetically different clones, we're talking cells with completely different mutations, could all end up switching on the exact same survival programs to fight off the treatment.
This idea is called convergence, and it pulls back the curtain on one of cancer's most secret and powerful strategies. Let's break down what this actually means, because this is where it gets really interesting. The idea is actually beautifully simple, but its implications are profound. So on one side, you have these different groups of cells with totally unique genetic mutations. Their blueprints are completely different. But on the other side, they all end up using the exact same survival strategy, like flipping on inflammation pathways to shield themselves from the drugs we're using.
This family tree shows it perfectly. Look, you can see these different evolutionary branches, each one driven by a totally separate mutation that helps it resist therapy. But even though they took different genetic roads, they all ended up at the same destination, this inflammatory state that helps the whole tumor survive.
So what's the big takeaway here?
It means cancer doesn't just have one trick up its sleeve. It has multiple, totally independent paths to arrive at the same winning move. It makes it a much smarter, much more resilient enemy than we ever gave it credit for. It's like a chess master that knows how to win the game from a dozen different positions on the board.
But the story doesn't end there, not even close. Remember that third superpower I mentioned? Well, this might just be the most revolutionary part of all. Okay, so imagine this. For decades, hospitals all over the world have been preserving patient tumor samples in little blocks of wax. It's a method called FFPE. We're talking millions upon millions of these samples, a literal medical time capsule. They hold the stories of countless patients and diseases, but the preservation process damaged the cells just enough that our old technologies couldn't read them.
Until now. This right here, this is the proof. This data is from an old archived wax-embedded sample that was analyzed successfully using GoT-Multi. For the very first time, researchers could look back in time and map out the entire complex world of cancer cells and immune cells from one of these preserved samples.
You have to understand, this is a total game changer. It's like we just found the key to a global library of clinical data that's been locked for half a century. Researchers can now go back to samples from patients treated 20 years ago and figure out why some responded to a drug and others didn't. We can study how diseases evolve on a scale that was just science fiction before.
Which leaves us with this final, really exciting question. By finally being able to read these incredibly detailed stories hidden in our medical past, it's not just about cancer anymore. What other complex diseases, things like autoimmune disorders or neurodegeneration, could we finally learn to outsmart? The possibilities are just beginning.